video is going to describe the chick embryo model building assignment. And the model that we're going to build is based on this image of a chick embryo. And within a single chick embryo, there's a range of development occurring. The development is more advanced further along in the more anterior region of the embryo, and then a little bit later in time in the posterior, and we'll talk a little bit about why that is. And so up in this region, the neural tube is forming, and the formation of the central nervous system from anterior, that's the head end, so the head forms first down to posterior. This is toward the tail end of the embryo. And then down in this most posterior region, we also have gastrulation occurring. So this is a great model to build because this is occurring in just one embryo, and we can look at a range of things. And so that's the goal of this video, is to introduce you to the assignment. So the next thing to say is that at this stage we look like this. So another reason for us to build this and emphasize this model is that humans look like this. And then finally, we're pretty close to the phylotypic stage. It would be a little bit more advanced than this, but pretty close to the phylotypic stage of all vertebrates. And so it's another reason for building this, is that we have a lot of features that are similar to other vertebrates. So I got some materials for building models like this outside of my office in the fourth floor of the new wing, and there are some tables nearby, and you're welcome to come over and use those materials. You're not expected to go out and buy anything. If you have some good materials like this, art supplies at home, go for it. And the idea of having supplies is to have some creativity. Creativity becomes memorable. So, if you use a color-coded paper for the endoderm, mesoderm, ectoderm, that works out, is helpful to help you as you're working with it to learn the structures, and then when you're reviewing it or remembering it, that creativity and that variety can help you as well. So there's stuff other stuff in that bag too, and people will build models that are somewhat distinct. You don't have to hand in your model. You can hand in pics of your model. Your model should be labeled, and it should be labeled, and it might look something like this. It doesn't have to look like that. You're not trying to create the image as sort of exactly as this student did in their model. But just to give you an idea that we're trying to make a 3D model, and it's based on that, and then we're labeling it like that. In addition to the labels that are in the figure, we should strive to add these labels that are found in all vertebrates at the vertebrate phylotypic stage. And we talked about some of them. I'll introduce a little bit more of them in this video, and we'll talk some more about them in class. And in our class, we'll eventually add a couple more features of the vertebrate phylotypic stage, but this is a great list for us to work on for this assignment. And so try to include these labels in your model as well. All right, and so this image is a relatively flat embryo. The chick embryo develops like a flat disc or a coin, and so we just want to talk a little bit also about frog embryos, because frogs develop as spheres, and the first thing we've started describing in our class, and the first thing that's described in the textbook in chapter 3, is a frog embryo. And so here's a frog embryo, and we see that the egg is a sphere, and the fertilized embryo develops as a sphere to start with. And so we first form blastula, and then gastrulation begins, and then something called neurulation occurs. And so we've talked some about how that the blastula is a hollow ball, and then a dent forms the blastopore, and then endoderm and mesoderm fold into the body through the blastopore. And once that occurs, we've got a notochord, and that's setting up induction of the neural tubes. That's called neurulation. And that's all happening as a spherical shape. So here, for example, is a spherical fate map. And you don't need to pay attention to this right now, but just the sphere for the frog embryo. Here's some drawings a student did of the blastula, the gastrula, and the neurula at a different year in our course, like that, just to give you, again, a feeling that it's a sphere. Here's this cross section that we talked some in class about to start learning about the ectoderm, mesoderm, and endoderm. It's a sphere. And again, we're going to be making a flattened model, a flattened disc. That's going to have some three-dimensional features to it, but it's more like a disc, like a coin, than a sphere. And the reason there's a difference between the sphere and the flat embryo is based on the differences in early vertebrate development. They're all converging on the phylotypic stage, but they're doing so through some different features. Here's a chicken egg, a chicken egg yolk, and the early embryonic disc develops in a region that's about that big relative to that chicken yolk. And here's a blow-up of it just under a regular light microscope. The central region where the embryo is called the area pellucida. Around it is this area called the area opaca. And what's going on there is that in a frog, when we have this sphere of the early embryo start to develop, the frog rapidly divides it up into a series of cells through a process called mitotic cleavage. We learned that there's more yolk in the lower part of the frog, the region of the vegetal pole, and as a result, the yolk, the sticky yolk, makes it so that the cleavage is uneven, and so we have smaller cells up here near the animal pole and larger yolk-filled cells down here at the vegetal pole. Nevertheless, the frog undergoes what's called complete mitotic cleavage. The chicken egg is just so gigantic. That's one chicken egg cell that's just been stocked full of nutrients and regulatory factors, maternal factors. And as a result, birds and reptiles have incomplete mitotic cleavage. And so what happens there is that it just can't cleave down into this yolk region, and so it forms a series of very small cells up on this surface that develop into a disc-like shape, like that. Okay? And you get a feeling for that transition from the amphibian that does have smaller cells and the animal pole, but nevertheless has complete mitotic cleavage, to the even larger eggs of birds and reptiles, and they just have incomplete cleavage. And so that explains why the early start of development in amphibians is different than birds and reptiles, and that they're going to converge toward this phylotypic stage. Okay? And so here we're looking down at this region on the chick egg surface, and on the fertilized egg it starts to develop. And here's this region called the area pellucida. That's where the embryo is going to be developing. And what happens is on one end of this circle, we start to see some development here in this region of the epiblast. If we looked at this in cross section, like we're looking at a coin where this is the top and the bottom, it says we see a thickening of this top region, which is called the epiblast. The lower part is called the hypoblast, and the hypoblast is going to make embryonic cells that are part of the extra embryonic membranes. So the embryonic sac that the little chick is developing in, and the allantois, which is like a gas exchange for oxygen and carbon dioxide. And so there's a thickening here that becomes a dent called the primitive streak. And if we did another cross section, the primitive streak really is a dent, like that. It's a little valley. And the primitive streak starts to form here, and it advances out like this. And eventually there's a bump at the end of the primitive streak, and that's called Henson's node in birds and reptiles. In mammals, it's just called the node. And then after that's forming, then primitive streak regression begins, where the node moves in this direction. And during that time, it's called primitive streak regression, and that's when gastrulation is occurring. So we're folding the embryo in together to get the endoderm, mesoderm, and ectoderm in their proper orientations. And when we do that, we're going to have cells proliferating in the node, sliding down into that groove and then getting covered up. And then we're also going to have cells sliding in from the sides of this valley. To try to draw it a little bit of an angle so you can see it. So here's the node, and here's that valley. And as the node is moving this way through primitive streak regression, it's moving from the eventual anterior end to the posterior end, and so the anterior end of the embryo is more advanced. And if we could see into the embryo here, we would see that the node has created three types of mesoderm. There's the notochord, and I'm drawing them as a dotted line because they're covered up. They were in this dent as they were forming, but then as the node moved over things, it left a smooth surface behind it. And because the notochord is there up at this end, we start to get neural ridges and eventually pinched off and a neural tube up here. So this anterior end is more advanced. The head neural tube that's going to produce the brain forms earlier than the more posterior spinal cord of the neural tube. So the first thing is that notochord is that notochord. Then flanking it are another, and I call these like the five fingers. So we have a middle finger, just as by analogy, because it's finger-like, and it's it's a tube or a hot dog-shaped thing. And then there are these two other hot dog-shaped things that are called somites, one on each side because of bilateral symmetry. And then flanking them further out, more fingers, hot dog-like structures, and those two would be the equivalent of like the pinky and the thumb. So we kind of have the five fingers getting folded in there. They're all mesoderm, and they are composed of notochord, then flanking it the somites, then flanking that the lateral plate mesoderm. And I've gotten this typed out in a slide. And so those are going to form notochord, somites, and lateral plate mesoderm. And so those are forming, and they're forming up here in a more advanced stage, and they haven't occurred yet down here because gastrulation is just beginning and ongoing there. Okay, so these fingers forming, and again, I'll call them fingers. They're not our fingers, of course. They're just have a similar shape like that. Okay, so this is showing that image here where this is the primitive streak. It's not depicting the node here, but the node is a little bit of bump. You can see the hypoblast, and then endoderm, mesoderm folding in, and the node up here is also going to be producing a lot of the mesoderm. And so that's going to be the process of gastrulation. This is just from a different textbook that shows that that the frog embryo develops as a sphere. So in cross-section, we see the notochord, the neural tube, the somites, and the lateral plate mesoderm. The chick normally develops as this more flattened disc, but what it was done here, this author decided to bend the sides of the coin to make it more similar in shape. So they're very similar in their organization. It's just a question of one being more of a flattened out version of the other. Okay, this just shows something in more detail, and I think we'll come back to that figure in a moment. So let's just look at this list. I want to just say something briefly about a couple of these. So we really will continue to talk about the notochord, but we've talked about that some. We've talked about the deuterostomes and the triple blast body layer in class, the ectoderm, mesoderm, endoderm. And this is a new term for us, lateral plate mesoderm, and you'll see that the lateral plate mesoderm gives rise to the splanchnoplura and the somatopleura. And we'll talk about that in the context of forming our chest cavity, our gut cavity, and that's called the coelom or coelomic cavity. That body cavity where you can flap open your chest and you can look at your internal organs because they're in your chest cavity, your body cavity. That's called the coelom or coelomic cavity. Then we've also talked about the developmental biologists call the digestive tube the gut. and we're deuterostomes, so we have a digestive tube. The official term for that we haven't mentioned in class, but it's mentioned in the book, is called the archenteron. And then we know we get a neural tube and neural crest cells. So some of these things we've already introduced the terms and a little bit about what they do. The main thing we want to talk a little bit more about are these pharyngeal pouches and the lateral plate mesoderm. So the pharyngeal pouches are in what will be our eventual throat region, and there are half a dozen pouches. They're called either the pharyngeal pouches or the branchial arches. Branchial arches are actually found in fully grown organisms, and they're the gills structure, the gill support in a fish. And so they are officially in an adult fish. Unfortunately, plenty of people refer to them as branchial arches in embryos, and that leads to confusion. You may have heard this idea that some people think when humans were embryos, we had gills, but we never did. It's a misunderstanding. Gills are differentiated tissue, mature cells in an organism like a fish, and sort of ancestral vertebrates that lived, millions of years ago. We don't have branchial arches. We have these pharyngeal pouches, or some people call these pharyngeal arches. And what they look like is this. They look like just little pouches. And the book doesn't talk about them very much. In lots of settings, they're talked about a lot because the pharyngeal pouches give rise to different components of the vertebrate lower head and throat. And lots of stuff happens here in the evolution of vertebrates. For example, really primitive vertebrates had no jaw. So they have a mouth, but they can't even chew. And there are only a couple of modern vertebrates, lampreys and hagfishes, that are jawless fish. But our ancestral vertebrate was that. So you go from something that didn't even have a jaw, and then part of these pharyngeal pouches give rise to the jawbone, and so forth, and all the features of the mouth and jaw. And then we have also really complicated stories about the inner ear and the bones of the inner ear, and how they've evolved during vertebrate evolution. So that inner ear has changed quite a bit. And then down in the throat, we have a lot of different features. Those have affected how the cranial nerves grow out from the hindbrain and innervate the face and the throat, and go down into the chest to innervate some targets down there. And we don't need to know all about that. I think our book tries to steer clear of it because it becomes pretty complicated in the story of the evolution of vertebrates. But they're there. They're in our throat up near the anterior region, and they're composed of both ectoderm, mesoderm, and endoderm, and play a role in the lower part of our face, like that. Okay? So just talking about that briefly, this shows the body plan of an early vertebrate. Ancestral vertebrates had a notochord that stayed with them for their whole life. They were very simple, like a little tadpole, sort of, like that, but that was the adult. And they had a mouth, but no jaw. They had these pharyngeal slits, or sort of precursors to the gills and branchial arches of fish, but they weren't that developed yet, like that. Yep, they had that dorsal hollow nerve cord that in us is the brain and spinal cord, like that. Okay, all right. So pharyngeal pouches, just to add to your understanding, we don't have to try to really create them on our model, but we could put a label on where they approximately are located. Great. Then I also want to talk about this lateral plate mesoderm, and you can see that here. And where the lateral plate mesoderm actually is in this figure is it's here. So let me go to my little drawing pad and draw what's going on here. So when we see vertebrate embryo in cross-section like that, we would have that digestive tube, which in the embryo is called the archenteron. And the archenteron has endoderm lining it. And then we're talking about these mesodermal structures today. We have the notochord, and induced by the notochord overlying it, I'll draw it in the same color because we can say maybe I'll do a different color. Let's do a different color. So this is going to be the neural tube, and this overlying cells here will be the ectoderm. So the notochord induces formation of the neural tube in all vertebrates, so we have that. And then what we're talking about new here are some mesodermal structures, the somites. And the somites, when you look along their length, are going to get basically sliced like a hot dog, like slicing a hot dog to feed to a little baby, sliced up into individual somites. And the individual somites are numbered from anterior to posterior, and they're going to give rise to all the muscles and body and bones of the body, and also some internal organs like that. Okay, so we have the somites, and then we also have these solid fingers here, the lateral plate mesoderm, the LPM, lateral plate mesoderm, and so bilaterally symmetric, and the lateral plate mesoderm, instead of getting sliced into chunks like this, the lateral plate mesoderm starts to hollow out. So they both become hollow, and then that hollow expands. So I'm just going to do that right on here. It expands like that. So that's the hollow center, and then we have cells like this. And of course, the same thing is happening over here, like that. So we get this hollowing out, and in the center of it, there still is that digestive tube, the digestive tube or archenteron. And you can see that's what's happening here is that this hollow region is going to become that chest cavity, that body cavity, that coelom or coelomic cavity. Okay, so the lateral plate mesoderm in our torso, our body region, creates this, the lateral plate mesoderm creates this hollowed out body cavity. And in that lateral plate mesoderm, there are two basic types of cells. So here's one type of cell, and these cells are called the somatopleura. And those are on the outside, so like when you cut your chest open and flap it open, on the outside of that flap would be the epidermis, the ectodermal epidermis, the skin. On the inner part of the flap would be the somatopleura. And the somatopleura is boring because it's just the lining of our chest cavity. On the other hand, these cells are very interesting. The inner part of these cells here, the lining here, these cells are referred to as the splanchnopleuron. And the splanchnopleuron gives rise to internal organs, like the heart and kidneys. And so that's where the precursor cells would be for the heart. And you can see the heart actually develops as a bilateral structure and coming together. And so quite a complicated development, but that's where those are. And again, in your model, you don't really need to create all these features clearly, but you should try to label where they are because these terms are important. So in your model, we'll try to have you label where those are. So there's a side view or a cross-sectional view through a typical vertebrate, and you see the notochord, the flanking somites, and then this developing lateral plate mesoderm, where these are the two hollowed out lateral plate mesoderm coming together to wrap around the endoderm to have the digestive tube run through their gut cavity. Okay, awesome. And so here I just put both these on here, and I'll just try to draw on here a little bit. So in this figure, it's depicting, see, the lateral plate mesoderm getting hollowed out here, and they're less hollowed out here because this is posterior, and they're not hollowed out here at all. And you don't have to depict all of that, but that's what's trying to be depicted by the artist in this. As we go anterior to posterior, we have more developed lateral plate mesoderm, and that's what's happening to it as it develops. Then we also have the endoderm, and the endoderm is a tube a digestive tube that in the chest area is open to the underlying yolk. And so there would be yolk there, and so we have a digestive tube, and later that yellow digestive tube gets closed off. There's the notochord, and the notochord is found behind Henson's node. So there's Henson's node. It's moving this way during primitive streak regression. And there's the primitive streak in front of it, and it's moving down here to cover it up. And behind it, the notochord is forming. So the notochord has formed here and is already inducing the neural tube. There are some somites, that's what's depicted here, and then the lateral plate mesoderm. So hopefully that just provides some better understanding of the landmarks and the layout. And you don't have to include every single one of these terms, but you should include most of them in your drawing. There's the somatic mesoderm and splanchnic mesoderm, for example, we're trying to represent. And so we're really, trying to get these features identified in your model that you build. And again, you're welcome to work with it with other students. I think it would be very convenient to come over to my office with other students and one or more other students and get that bag of stuff and set it up on that table and just make yours at the same time as other students. And you have to hand in your own model, but it can be the same as others like that. Okay? And so that's going to be the end of our video for that. Bring questions that you have to class about this assignment. Thanks.